Hello, and welcome to Virtual Investor Conferences. My name is Matt Lateplo, and on behalf of OTC Markets, we're very pleased you have joined us for our next live presentation from ANGLE PLC. Please note you can submit questions in the box to the left of the slides. You can also view a company's availability for one-on-one meetings through the Schedule Meetings tab found on the conference platform. At this point, I'm very pleased to welcome Andrew Newland, Chief Executive Officer, and Ian Griffiths, Chief Financial Officer of ANGLE PLC, which trades on the OTCQX Best Market under the symbol ANPCY and on the London Stock Exchange under the symbol AGL. Welcome, Andrew and Ian.
Thank you very much indeed, Matt. And I'm Andrew Newland, and I'm going to explain to you how this small piece of plastic that I'm holding in my hand is going to transform the way that cancer is diagnosed and treated across all different cancers. And I know that sounds a very bold claim, so I'm going to back that up with a presentation that explains how this system works and how it's been validated and how we hope it's going to be adopted in the future. The background to cancer is that it's different between different patients. So one kind of imagines that if 3 women have breast cancer, then they all have the same cancer and they'll all be treated the same way. But in reality, that's not the case. And that's why, as standard of care, when a patient is admitted for potential cancer, they do a tissue biopsy. Now, the purpose of a tissue biopsy is they cut out some of the cancer and they take that material to a laboratory and examine it. And there are 2 real questions that are asked. The first one is, can we confirm the presence of cancer, in which case that's a confirmed diagnosis. And the second question is to examine the cancer cells and seek to find out information about the individual's cancer so that their treatment can be personalized. And this applies across all different cancer types. So I often refer to breast cancer as an example, but it equally well applies to prostate cancer, lung cancer, colorectal cancer, melanoma, brain cancer, and on. So all solid tumor cancers equally have the same information. So the issue, therefore, with the tissue biopsy is to determine how best to treat the patient. And that's all very well, except for the second major problem with cancer, which is that it changes over time. The cancer mutates in order to evade the immune system and evade the drug therapies. So after a bit of time, then the cancer is actually quite different oftentimes from what it was when the patient first presented. So that causes some significant problems. And the need really is to be able to repeat a biopsy so that the up-to-date information can be obtained on how the cancer is progressing and which therapies would be most appropriate for the patient at that point in time. The difficulty is that but at that point in time, it's not possible to repeat the tissue biopsy in most cases because the cancer's already been cut out. And in my breast cancer example, the patient may have had a lumpectomy or a mastectomy. So you can't go back and recover some more cancer cells from that point for the purposes of analysis. So there's a need for a new modality, a new methodology by which to get cancer cells for repeat biopsies. And that's what we've developed with our Parsortic system that's shown in this first slide. So this instrument is designed to recover cancer cells, so we call it harvesting, to harvest cancer cells from the patient's peripheral blood for subsequent analysis. So why does a patient who has breast cancer, as my example, have cancer cells in their blood? Well, that's because that's how the cancer spreads within the patient. So cancer cells leave the primary tumor, in this case the breast, and then they go into the blood, and these cancer cells are then called circulating tumor cells, abbreviated to CTCs. So circulating tumor cells then go round in the development. That process is called metastasis. So the process by which cancer cells leave the primary, end up in another organ and start growing is the metastatic spread caused by circulating tumor cells. So there is actually, therefore, an available source of up-to-date cancer cells in the patient blood. which if they could be recovered, would be a perfect source of information on exactly what the cancer is doing. And by the way, to add another little bit of color to this, if you look at the mortality rates for patients with cancer, in fact, well over 90% die from the secondary cancers, not from the primary. So if a patient dies of breast cancer, it's generally because it's spread to the lungs and the liver and the bone and the brain as secondary cancers. And the way that it gets there is by these circulating tumor cells in the bloodstream. So they're very, very clinically significant cells. So what I've told you so far is sort of very well understood. In fact, it's been known for maybe 100 years that there are these cancer cells in the blood. But there is a very big technical challenge with accessing them, which is that they're very rare in number. So there might be, in the picture here, you've got a tube of blood. That's a standard blood tube that the peripheral blood draw can be drawn into. It's a 10 milliliter tube, but there's probably 7 or 8 milliliters of blood in this particular picture. And that will constitute probably as many as 8 billion, so 8,000 million blood cells, most of them being red blood cells, but many multiples of hundreds of millions of white blood cells as well. And inside that, there might be only 5 or 10 cancer cells. In a very progressive patient, there might be several hundreds, but not that much more than that. And in extremis, there might only be 1 cancer cell there. So they're rare events, and it's very, very difficult to reliably capture them. And what I showed you a second ago in terms of the piece of plastic, that is the Parsortix cassette. It's a one-time use microfluidic device that we have developed within Angle. It's proprietary and patented and owned by Angle. And we can flow blood inside one of these Parsortix cassettes using the instrument shown in the picture and recover the cancer cells very, very reliably. So reliably, in fact, that as you can see on this slide, we have an FDA product clearance for the intended use of metastatic breast cancer, recovering these cells. So that's a bit of background. Now, the issue that we're addressing is a very, very widespread issue as to how to get a good sample for analysis. Now, the beauty of the sample that Angle's providing is that it is intact, living cancer cells which have been recovered at that time point from the patient. And it allows all of the full analysis of the cancer to be undertaken on those cells. So it's possible to do DNA, RNA, protein expression, and also morphology analysis on those cells. We believe it offers the best possible sample source for doing repeat biopsies in cancer. Now, the way that the cancer is developing is through clonal evolution. So when you hear a story that a friend or a family member has got cancer and the treatment is going very, very well, what they're generally referring to is a reduction in the size of the primary tumor. Unfortunately, what will then happen sometimes is that you'll get the message that, oh my goodness, it's suddenly started expanding and spreading again. Well, of course, the drug that the patient's having hasn't changed, but the cancer has changed. The clonal evolution of the cancer means it is now able to avoid that therapy, and a new therapy decision needs to be taken. That decision at the moment is driven, broadly speaking, on guesswork because there is no new information on how the cancer has changed. And that is what we're seeking to address with our Parsortix system. And that's a well understood requirement. In fact, the National Cancer Guidelines in the US for breast cancer patients state that if the patient has a secondary cancer in breast cancer, so for example, the lungs, the liver, the bone or the brain, as I mentioned before, then they will attempt a biopsy of that site to recover cancer cells to investigate them to guide treatment. And that involves the patient having to have a general anesthetic and full surgery. For example, if it's spread to the liver, they're cutting out part of the patient's liver, not to improve the outcome, but simply to find out information about the cancer. And in fact, most patients, over half of patients, don't get this second biopsy because they're too sick or the cancer is inaccessible. So what we're offering is a simple blood test approach, and it wouldn't just be done once, as in the guidelines. It could be done every few months or so. So this is to show you in this slide how the technology works. We have got the, this is just a schematic of the Parsortix cassette that I held up. The blood flows in the inlet, it flows down the microfluidic channel, but you can see it's closed at the end. So it has to throw left or right. And that takes it up a cross section with a series of steps. And those steps are set so that the red and white blood cells, which are significantly smaller and more compressible than the cancer cells, can go through. But the cancer cell is larger and less compressible. It doesn't make it through the critical gap. So all the machine does is to automate the flow of the blood through this cassette, allowing all the blood cell components to flow through, but the cancer cells to be held gently in the Parsortix cassette. And the machine is very cleverly automated so that it's very simple to use. All the user has to do is to take the top off a standard tube of blood, attach it to our instrument and hit go. And the Parsortix instrument will do all of the work. It will sense when the blood sample has completed. It will then automatically put a saline wash behind to wash away the remnants of the blood. And then once that's complete, it will then reverse flow and flow back out the cancer cells separated from the blood cellular components. And that provides the cancer cells alive, undamaged, and in a form where they can be easily analyzed for a variety of different methodologies of investigation. So having developed this fantastic system and got it through FDA regulatory approval, which was a pretty significant challenge. We took 6 years to do that and had to process 16,000 samples and develop 400 technical reports to meet FDA requirements. That has now given it a gold standard stamp of approval, and we are seeking to commercialize via two businesses, a products business and a service business. So the products business involves us having distributors in multiple geographic territories where we're seeking distributors who have got basically customers who buy oncology related product, and they're now selling our Parsortix system. And we've got these distributors across Europe, in the Middle East and in the Far East. We currently sell in the UK and in the United States and North America generally via direct sales, but we will shortly be moving the North American side into a distributor as well. So here what we're doing is we're selling the instruments as widely as possible to research and clinical labs who can then use the cells that come out for a variety of different downstream uses. And because of the way that we have a consumable associated with this, it's effectively a razor blade model. So we sell the instrument and then we sell the Parsortix cassette as a consumable for each time that the system is used. So that's the products business. The services business involves us having our own clinical laboratory here in the UK, which is running clinical lab quality standard procedures such that it can take blood. So the customer can have the blood sent to our laboratory. We have a bank of our Parsortix machines. We then process the blood and recover the cancer cells, and then we can do a variety of different analyses for the customer. Now, this business is taking off now with our pharmaceutical customer base because the pharma companies can now have the patient's blood drawn before the patient joins their trial, and the status of the patient can be assessed by looking at their blood, and then they can have their blood taken again at the time of them having their drug in the trial, and then again afterwards several times to see whether any response is maintained. So the whole idea here is that you can give the pharma company a lot of information about how an individual patient is responding or not responding to their drug. You can also look at the cancer cells for the target that the pharma company is looking for in their specific clinical trial. And this is, again, it's all to do with personalized medicine. The new drug treatments which are coming through are increasingly personalized for subsets of patients who will respond very, very well. It's interesting statistic that if you look at immunotherapies, which are essentially very important new drug category for cancer patients, these drugs stimulate the patient's own immune system to kill the cancer. They're very expensive, so they can cost $150,000 to $200,000 for each patient, and yet they only work for 1 in 5 patients. So 4 in 5 patients, that funding is lost money, and the patient unfortunately will receive toxic side effects from the drug and yet has no benefit. So clearly it would be extremely helpful if you could have a blood test to work out which of the 5 is likely to respond and which 4 should in fact be given an alternative drug instead. So now that we've got the system for recovering the cancer cells, we are working on content as to what you can do with the cancer cells once you've got them. We're just about to launch a new product called Portrait Plus, which is a kit which provides antibody staining for the cells. So now we will be selling a kit that the customer can buy with all of the antibodies optimized for staining the cells to characterize them as to whether they're epithelial cancer cells, mesenchymal cancer cells, or EMT cancer cells, and enable them to be assessed and enumerated and looked at under immunofluorescent microscope. So by doing this, we're making it as easy as possible for our customers to adopt our technology, because now we've given them the full instructions all the way through to the endpoint to assess what's there in the sample. We're also working on a particular protein analysis called Herto. In the short presentation like this, I don't have enough time to explain it fully, but it is a key target in breast cancer. And there are drugs which target HER2 positive and HER2 low patients. So, and the HER2 status can change. So here what we're doing is identifying the cancer cells and then looking on the cancer cells as to whether this key protein is expressed or not. And then from that, you can deduce whether or not the patient is likely to respond to a particular drug. We're also working on content in relation to molecular analysis. I'm sure everybody in the audience has heard of the Human Genome Project and DNA sequencing of cancer to identify different mutations which may be targets for drugs. And in fact, there are now multiple drugs which are FDA cleared for patients with particular DNA status. So if they have a particular DNA variant which is identified, then they will be given the drug, but they won't if they're not. There's an alternative method for liquid biopsy which is widely used, which is circulating tumor DNA. Now that is fragments of dead cancer cells found in the blood. So that contrasts with the living cancer cells which are intact, which are recovered by Angle's Parsortix system. But what we've developed is a molecular process using industry standard Illumina sequencing to measure the DNA variant status on both the circulating tumor DNA and the circulating tumor cells DNA from the same tube of blood. And this is in this chart here, some early data in relation to that. And what you see here is the circulating tumor DNA information. And where there's a number, that means that that mutation is present in the circulating tumor DNA. So if you look at that particular one, EZH2, that is present both in the circulating tumor DNA and in the circulating tumor cells, both in the dying cancer cells and in the living cancer cells. But by doing this analysis, you find some very interesting anomalies. So for example, there are some mutations which are shown just in the circulating tumor DNA. So that's where it's in the dying or dead cancer cells, but it's not in the living ones. That perhaps suggests that the immune system or the therapy has sufficiently dealt with that brand of the cancer that it's no longer in the living component, it's just in the dead component. And then, as I said, you've got some where it's in both, so that implies that the therapy is working but it's not yet completed its job. And then very interestingly, and this is new information, in some mutations, we're finding that they're positive in the circulating tumor cells, but not in the dead circulating tumor DNA for this particular patient. So this is a patient with small cell lung cancer, and this particular patient is EGFR mutant in the living cancer cells, but there's no sign of that in the dead cells. And that would suggest that this is part of the clonal evolution of how this particular patient's cancer is evading the drug therapies that are being offered for this patient. So had the oncologist had access to this information, they might have said, well, that's very interesting. What can I do about it? Answer, there's a whole series of FDA cleared drugs which target EGFR mutant cancer in lung cancer patients. So there are prescriptions that could be given. There's another one down here, PIK3CA. There are also PIK3CA inhibitors, FDA cleared, that could be used here. And again, it's in the circulating tumor cells, but not in the circulating tumor DNA. So that content and the marrying up of our Parsortix system with industry standard sequencing solutions, we believe is going to drive very widespread adoption of our technology to produce the sample for analysis. Now, in our field, it's critical to have third-party independent corroboration of the ability for technologies to do different things. And in our case, we've widely distributed our instrument to very, very leading cancer centers. In fact, we've got 39 world-class cancer centers spread around the world, United States, Europe, and elsewhere. And these 39 cancer centers between them have generated actually it's now over 90 peer-reviewed publications that have gone into leading journals, every single one of which shows that the Parsortix system is reliable and provides a good sample for downstream analysis. And in fact, whilst I talked about breast cancer particularly, they've shown that it works in 24 different cancer types without any modification of the instrument or the consumable. So we think that sets the company up for a very widespread adoption of our technology. And this is my final slide. So we've got a number of commercial milestones which are pretty much imminent. We're building out our pharma services business, and we're expecting to be able to announce some new contracts fairly soon, including with some of the world's largest pharmaceutical companies to use this technology in their trials. We're rolling out the new assays. I haven't mentioned it in this presentation, but we've recently launched Portrait PDL1 as a service. PDL1, a bit like Herto, is a protein which is a target, in this case for immunotherapy. We've also got DNA damage repair assays in our clinical laboratory as well. I mentioned about the Herto. We're launching this Portrait Plus kit next week at the San Antonio Breast Cancer Symposium. The molecular information I showed you with ctDNA and ctcDNA, a lot of work is going on that to build out the data sets. And we're hopeful that we're going to get some announcements on some pretty major breakthrough information on this molecular readouts pretty soon. And finally, we've got several major clinical studies, not just breast cancer, but also in ovarian cancer and prostate cancer, which will read out in the near term. So with that, I'd like to ask Ian to join me, and then we can go to questions. And just as a first point, we're open to answer any questions that you may have. If you want to send them in to those email addresses on this particular slide, and we can potentially set up some follow-up meetings as well if there's interest. Thank you very much. Ian, do you want to take over on the questions?
Yes, thanks. And the first question that came in is, which countries is Parsortix approved for sale in?
Oh, that's a good question. So the FDA clearance, of course, is in relation to the United States. We also have a matched clearance called the CE mark covering Europe. And we have a MHRA clearance covering the UK. And that is for the intended use in breast cancer. We're selling and we're exploring, we're progressing some clearances in some of the other countries in Far East. It's very important to note, however, that we can sell this system for research use to clinics and centres all over the world. So we're not restricted in that for research use.
Thank you. Next question that came in is, are you still on target to publish the first third-party molecular analysis by the year end?
We're very hopeful that we'll be able to publish some headline results from that by the year end, yes.
This one partly was covered in the presentation. It says, Angle have previously indicated that they are in discussions with a number of major pharma companies. Are these talks progressing as anticipated?
I'd say that the market in our pharma services business has been challenging. So many, or the largest proportion of companies developing new drugs are what you might call biopharma. And they are companies that have raised private equity, or they've listed on NASDAQ or elsewhere to raise capital in order to invest in bringing forward new drugs. These companies have been hit by a very adverse funding environment at the moment, with the availability of capital being constrained. So unfortunately, that's fed through to us to an extent, whereby customers that we were about to start studies with have told us that, well, we still want to work with you, but we're postponing this study in the current market until the availability to capital improves. In response to that, we have moved our proactive efforts heavily towards the large pharma. Now, they're very different from biopharma, because the large pharma have billions or hundreds of billions of dollars of revenue, and so they don't have the same cash constraints. Their sales process is somewhat longer, though, so signing them up takes a bit longer, but we remain on track to sign some large pharmaceutical companies who could offer us potentially hundreds of millions of dollars of revenue just from a single client.
Great. And the next question is, ctDNA is well established in the market. Why would you want to change to CTCs, and can you explain the difference a bit more?
Yes, so ctDNA, which is the fragments of dead cells, the reason that's there is because our bodies are made up of billions of cells, and at any point in time, some cells are dying and there are new ones growing. So the old cells have to be got rid of. So they fragment into millions of tiny little fragments and go in the bloodstream, and then they are excreted from the body. So for a short period of time, they exist in the form of cell-free DNA in the bloodstream. If the patient has cancer, they may have a small subset, maybe 1%, of their cell-free DNA coming from tumor, which would be circulating tumor DNA. And that sits in the plasma liquid component of the blood. Anyway, long and short is, it's very easy to get hold of the plasma. So centrifuge the blood, remove the blood cells, and just take the liquid and sequence that. The difficulty is that, first of all, it is dead. And as I tried to point out in the earlier slide, there's a lot of extra understanding that can come from living cells versus dead cells. And the second thing is that because of its fragmented nature, and the fact that it's, well, just mainly because it's fragmented, you can only do DNA. You cannot do RNA or protein expression or morphological analysis. So the circulating tumor cells are additive to ctDNA. And our aim in showing the Parsortix BioDetect solution that I described in the slide is to encourage ctDNA companies to actually adopt CTCs as well. It's not instead of, but as well. And you can get both analytes very easily from the same tube of blood, because the waste product from ctDNA is the cells, and you can put them back into some liquid, run it through Parsortix, and you get the cancer cells out. So we're encouraging the adoption of both analytes.
Cool, thank you. The next question I've got is, if you're able to identify treatment targets through third-party sequencing platforms, will this enable clinical use, or will you need to run specific clinical trials?
So the first step is to show that we can marry up the Parsortix system with the sequencing platform and get useful information out of it. That is expected to drive a widespread research use, both in academic research, cancer researchers, and in pharmaceutical companies who are developing new drugs. The next element is to get it adopted in clinical practice. And once we've got a solution in our laboratory that we're happy is fully validated and we're operating the right quality control systems and accredited in the lab, we can then offer a clinical test to identify the presence or absence of key variants in the patient's cancer. That is distinct from us making a claim that if we find the EGFR variant, then the patient should be given XYZ drug. If we want to make that claim, we would have to run a clinical study to show that the patient benefited from that drug. But we don't have to do that. We can simply say that that variant is present, and the oncologist can then make the decision themselves based on the published data as to which of the FDA-cleared drugs that they might want to prescribe. We anticipate, however, that in due course, the pharma companies will actually want to get a full clinical study evaluating the use of their own drug with a particular status. And so they would then pay for any of those kinds of trials, and it would generate revenue for Angle.
Okay, I do have a number of other questions, but I am conscious at the time at 30, so we'll endeavour to answer those individually at a later point. So thanks very much.